Good afternoon, maybe evening, everyone. It's my pleasure to welcome you, both the audience here in the room, but also online, to the Sir Geoffrey Wilkinson Lecture in Chemistry. This is a flagship lecture for the college. Annual lectures like these are a unique opportunity to come together as a community, connect with international researchers, and to welcome alumni and friends back to the college. So thanks for joining us. We're delighted to be hosting you here at the White City Deep Tech campus, which is at the heart of Imperial's West Tech corridor. Our vision is to use this district as a powerful engine for attracting new businesses and investment to the area, hoping to drive growth across London and the wider UK. Already, academics here are publishing groundbreaking research and turning these findings into exciting new ventures. Alongside this, businesses large and small are leading the world in areas such as quantum engineering, clean energy, and machine learning. So before we get started, some quick housekeeping. For everyone in the room, there are no planned fire alarms this evening. So in case of alarm, please do evacuate. If you follow the emergency exit signs, either out the doors at the back or front of the auditorium, and assemble at the North Campus car park, which is the one by GradPad. So we're very proud to be able to acknowledge the legacy of Professor Sir Geoffrey Wilkinson through this event. Sir Geoffrey was an alumnus of Imperial Chemistry who went on to become one of its youngest professors for the time. His research was vast and pioneering, but he's perhaps most often celebrated for determining the structure of ferrocene and pioneering organometallic chemistry. These achievements earned him many accolades, including the Nobel Prize in 1973. We're deeply grateful to his family and friends at the Wilkinson Charitable Foundation who have supported the department and made this lecture series possible. It's our pleasure to celebrate his legacy by welcoming the pioneering chemists of today to share their work and inspire us all. This year, it's my privilege to introduce as our lecturer, Professor Stuart Schreiber, often described as the father of chemical biology. Professor Schreiber's roots are in classic synthetic organic chemistry. He received his PhD from Harvard in 1981, working on the oxidation of tertiary amines under the supervision of Professor Robert Woodward and Professor Yoshito Kishi. From there, he was directly appointed assistant professor at Yale, where he stayed on until 1988, when, having quickly transitioned through the professorial ranks, he moved to Harvard, becoming the Morris Loeb Professor in the Department of Chemistry and Chemical Biology, a position he still holds today, alongside being founding Core Institute member emeritus of the Broad Institute of MIT and Harvard, and founding CEO of the new institute, Reno Bioworks. Professor Schreiber is also a member of the National Academy of Sciences, the National Academy of Medicine, and the American Academy of Arts and Scientists, and has been recognized by many prestigious awards, including the Arthur C. Cope Award and the Wolf Prize in Chemistry. In the early stages of his academic career, Professor Schreiber distinguished himself through the synthesis of incredibly complex organic molecules, culminating in the synthesis of the immunosuppressant FK506. It was his efforts to understand the workings of this therapeutic that led him to the seminal discovery of a new mechanism of action for drugs, as so-called molecular glues, about which we're going to hear more in his talk today. Working together with Professor Jerry Crabtree, these molecules allowed him to map the first membrane-to-nucleus signaling pathway, that of calcium-calcineurin NFAT, and concurrent with Professor David Sabatini, to discover mTOR and the wider mTOR-dependent nutrient response signaling network. Professor Schreiber's creative use of chemistry has led to many other seminal discoveries. For example, his lab is also known for its discovery of the long sought-after first histone deacetylase, HDAC1, and alongside the labs of Professor David Alice and Michael Grinstein, their work demonstrating the role of chromatin marks in gene expression. He also pioneered the development of diversity-oriented synthesis, and the application of which has led to the discovery of many promising therapeutic agents. Beyond his own research, Professor Schreiber has had a huge influence on how we all do science. He was co-founder of the Broad Institute of Harvard and MIT, and as a serial entrepreneur, has been involved in the founding of more than 12 companies, including Vertex and Ariad Pharmaceuticals. During the COVID pandemic, he co-founded Stop COVID-19, a non-partisan science-based group who advised US policymakers on the global pandemic. Most recently, in 2023, Professor Schreiber co-founded Reno Bioworks, an exciting new biomedical research institute funded by private investors that aims to revolutionize drug discovery. When writing this introduction, it was difficult to select what to mention from amongst Professor Schreiber's many pioneering scientific contributions. Indeed, already back in 2016, when awarded the Wolf Prize, his contributions to chemistry were described as of once-in-a-generation magnitude. And yet he continues to make many more scientific and entrepreneurial contributions to this day. So I think that's enough from me. It's now time to give the floor to Professor Schreiber, who's going to tell us more about the burgeoning field of molecular glues. So over to you. I look forward to your talk.
Thank you, Louise, and thank you all for coming. It's a real pleasure to be here today. Louise, what you were describing, this campus, it's the first time I've been on this campus. I'd been to Imperial College a couple of decades earlier, and it's exactly as you described. It's been really exciting to see the kinds of science that's being done here, the entrepreneurial spirit of the place. And especially I'm grateful to the trainees who spent their time with me today. And boy, did you leave me with a very strong impression of the quality of students and postdocs here. So now I thought I would, if you don't mind, I'm going to start with a little personal story that's not directly related to what I'm going to talk about today, but it reflects why it's also very special for me to be here to honor Sir Geoffrey Wilkinson. So, why do I feel a connection? When I was very young, and I went to college at the University of Virginia, I met an advisor, and I told him I don't really know what I want to do, but something in the natural sciences. And he said, okay, then you start with chemistry. I said, fine. And then he told me, he looked at my records, he said, but I'm sorry you're not qualified, because it was true I had never had high school chemistry. I tried some other things. It was not for me, and I quit college. A little bit later, I ran into my loving sister, and she asked me, why had I done this? And I told her the story I just told you. And she said, look, I support you, anything you want to do, but the one thing you can never do is listen to people tell you that you can't do something. And it was really that moment that I decided to go back to the college, and I entered into a large auditorium, something like this, and I entered in and I saw Professor Russell Grimes was at the board, and he was using blue and white chalk, and he was drawing to me the most beautiful art objects I had ever seen. I had absolutely no idea what they were. It turns out they were d orbitals. So now you can begin to imagine why these would be so appealing, right? And so that's the moment that changed my life completely. I flipped everything around. I came back to college, and I decided I wanted to be an inorganic chemist. And these d orbitals were always sort of driving what I was thinking. So I proceeded. It turns out that the next course you take is organic chemistry, and I fell in love with stereochemistry and conformational analysis and things of this sort, but never lost my sights on inorganic chemistry like Sir Geoffrey. I decided I wanted to work in my graduate studies with a famous inorganic professor at Stanford. I went out and visited. I told him why I love this field so much, and he said, I'm sorry, but actually we're not going to admit you to the program. And I said, why is that? And he said, you're insufficiently trained. He said something like, you're from Virginia and you'd have to be competing with these California trained students, and they're just like vastly superior. So my next attempt to follow Sir Geoffrey Wilkinson's footsteps, we're foiled again, but I did get into Harvard, and I joined R.B. Woodward's laboratory, and I got to work with him on writing a proposal, in which case I had this opportunity to tell him the story I just told you. And he said, oh, well, this is very interesting. He said, he introduced me to Sir Geoffrey Wilkinson. And I think Wilkinson was a member of the Society of Fellows at Harvard, a young scientist. And he told me about the origins of the structure of ferrocene. He told me a lot about Wilkinson, and this was, very, very inspiring again. In my PhD studies, I studied with Woodward iron and copper mediated synthesis. I went to Yale and then back to Harvard, doing organic synthesis, but always with a heavy reliance on organometallic chemistry. In the late 1980s and early 90s, I published a paper that I doubt any of you have ever read, but it is one of my all-time favorites, if not the top paper. Its title was Pi Bond Hybridization in Transition Metals, a Stereoelectronic Model of Conformational Analysis. And if you do go back and read that paper, you'll see that it was all about thinking about where these d orbitals are in these metals and how they interact with organic ligands. Now, finally I'm on the path that I wanted, and then something happened. It was a chance discovery. It was a chance discovery in a very different direction. And it's that one that caused me to move to chemical biology. And it's that story I wish to share with you today, the story about molecular glues. So now I'm pivoting entirely, except I'm also going to ask you to go back in time to that same late 1980s. There was a fascinating problem, very different kind of problem, called signal transduction. So by this I mean, how is it that an extracellular protein can bind the surface of a cell, never enter into the cell, and yet influence a series of intracellular events all the way into the nucleus, where a specific gene is activated and another specific gene is repressed? That was mind-boggling that this could happen. Now, let's see, there we go. So by this point of time, although the details were missing, the outline of how this might occur was beginning to be illuminated. And it was clear that protein-induced proximity was playing an outsized role. That a protein acts like a glue, and it binds an enzyme, and it binds a substrate or neo-substrate. And by a principle very familiar to chemists of high effective molarity, increases the rate of chemistry. And it's that process of induced proximity that occurs over and over and over as the signal is propagated into the cell. The chance discovery was in my lab, where we found that chemicals can induce proximity in exactly the same way, chemical-induced proximity. And we called those molecular glues. And so I think it's these sort of two pieces of input that catalyze a new way for organic chemists to think about how to intervene in biological processes by chemical-induced proximity. So that chance discovery is outlined here. Several trainees in my lab, led by postdoc Jun Liu, was studying the mechanism of action of these two natural products, cyclosporin and FK506. They discovered that despite their very different structures, they target the same protein, a protein phosphatase named calcineurin. But they do so in a very unusual way. These compounds first bind their presenter proteins, cyclophilin and FKBP12. Then those complexes bind cooperatively to calcineurin. They don't bind in the enzyme active site, shown here in the little asterisk. They actually block the entry portal for some substrates, but not all substrates. So they're substrate selective inhibitors. All of this seemed rather remarkable, and maybe something that was just a sort of a one-off, that maybe it was a billion years of natural selection, because these are natural products, that led to this amazing mechanism. That idea was disabused in my mind 10 years later when a graduate student named Steve Haggerty discovered the compound shown, we called it SynStab-A, or synthetic stabilizer. The two compounds, cyclosporin and FK506, we called molecular glues. This compound we called a molecular clamp, because in contrast to the former, it brings together two proteins that normally dynamically, weakly but dynamically interact, called alpha and beta tubulin. And they clamp them, they glue them together like a clamp. Now, this is exactly what the very complicated natural product Taxol does, among others. But it was a simple off-the-shelf chemical that's doing the same phenomena. And that's when we realized this may be very common, not very rare. Maybe it's everywhere. And I think today, many people would recognize that molecular glues are everywhere. We just didn't know to look for them. We knew that a third natural product called rapamycin was also a glue. It took another trainee graduate student, Eric Brown, to join my lab to figure out its mechanism. And again, it uses FKBP12 as its presenter. Together, they bind cooperatively to what was then an unknown protein. This is how the mTOR protein was discovered. We called FKBP12 the target of rapamycin, and we called this new protein the mechanistic target of rapamycin, or mTOR. And it works by the same mechanism. It's substrate selective. It blocks the entry portal for some substrates, but not all substrates. And then lo and behold, like 30 years later, now we have a beautiful paper from Felix Hausch, who synthesized simply a collection of about 1,000 compounds, simple synthetic compounds, and screened them, and he found a synthetic equivalent of rapamycin. It's a molecular glue for FKBP12 and mTOR, suggesting again that these are really quite common. And you can see the structure of the Hausch compound. So I want to begin with molecular glues. These are compounds that induce these ternary and higher order complexes. They do so cooperatively. This is key. I'll come back to this many times. And then I'm going to move to a second class of compounds. We call both of these chemical inducers of proximity, but I'm going to talk about bifunctional compounds that tend to have two elements that bind their target presenter or target independently, and then we simply connect them up with a linker. Those are called bifunctional compounds. You don't get the cooperativity out of them, but they're very appealing today because of this conceptual ease of discovering them. Find a binder, link them up. And I'm going to end today by showing you a new technique that allows you to discover either molecular glues or bifunctional compounds directly and by pre-selecting the presenter and the target of interest. So what I want to do is to begin with some very general principles. I want to frame all of this and give you a conceptual way to think about these compounds. So at the highest level, when small molecules bind dynamic proteins, they change their biophysical features. And I like to think of this small molecule protein complex as a neoprotein, creating like a new protein-like entity. These altered biophysical features are the basis of the frequent change in the interactome, i.e. a glue-like property. Okay, so first general principle. Jim Wells taught us almost 3 decades ago that compounds don't bind randomly to proteins. They bind to evolutionarily conserved regions of proteins he called hotspots. And these are regions that are normally involved in protein-protein interactions. So that was very eye-opening. The first new idea I want to share with you is the analogy of small molecule glues to missense mutations enshrined by natural selection. So missense mutation, I mean mutations that change the identity of an amino acid at a particular codon. Enshrined by natural selection just means mutations, as far as we know, occur rather randomly in the genome. It's the ones that we know about that had a function, and so they get enriched and we can learn and study about them. Now, what's amazing about missense mutations is that they do not appear randomly. The ones enshrined by natural selection are in or around those same hotspots. Second observation is that they function by changing the interactome. A missense mutation causes a new protein to interact that wouldn't normally bind. And this is, to me, is very appealing. You have a hotspot, you change amino acid residue, and suddenly a new protein interacts. Or you have the native hotspot, and you have a small molecule mimicking an amino acid side chain, and now neoprotein interactions occur. Lots of examples. This is one, just if you're interested, you get the reference from Haiyan Fu. He actually systematically studied the missense mutations we know about from somatic alterations in oncogenes, changes in amino acids that cause a protein to become oncogenic. And he set up a protein-protein interaction map, and he showed that over and over, these famous mutations induce neoprotein interactions, and they are biologically relevant. They explain the oncogenic mechanism of these missense mutations. Now, this is a more subtle example, but I love this one. It comes from researchers at Novartis. They were studying a paracaspase enzyme called MALT1. They were studying it because a mutation in MALT1 causes a Mendelian disorder. Single mutation, 100% penetrance, you will have this disease, a neurologic disease. And so they discovered a compound that binds an allosteric site of MALT1, and that's shown in the middle panel. But what they noticed was that in binding, comparing to the native structure, which is on the left, the native structure has an indole ring of tryptophan 580, and that's acting as a glue. If you look at the overall structure, you can see that tryptophan is taking two domains and gluing them together. They notice that their compound binds in a way that causes the indole ring to swing away, and their compound is a diaza indole. And that indole ring is sitting exactly where the indole, or diaza indole, where the normal indole of tryptophan 580 is. That gave them the idea of using patient-derived cells, because it turns out the disease I referred to is caused by a mutation of tryptophan 580 to serine. Not having this glue, that's an unstable protein, and this is the cause of the disease. So they wondered if this could be a molecular prosthetic, mimicking the natural amino acid side chain, and that's exactly what they found. They could take cells derived from humans with this disease, add their compound back, and it glued the two domains together and rescued the phenotype. Now, the second example that I want to give you, the analogy, is that of post-translational modifications. Take you back to the signal transduction. Here's an example of a cytokine outside of the cell binding its receptor, but with 2-to-1 stoichiometry. That causes the intracellular domains to get near each other, including the kinase domains where they transphosphorylate each other. High effective molarity, induced chemistry taking place, and putting a phosphate residue on a tyrosine. Well, there's a lot of proteins in the cells that transduce signals that have phosphotyrosine binding domains, such as lipid kinases. So when they bind this newly phosphorylated site, the lipid kinase is now in close proximity to its lipid substrate in the membrane. So this is just two steps, and over and over and over and over. High effective molarity, increased rate of chemistry, rendering these reactions now essentially intramolecular, and information being transduced. All the way into the nucleus, where, and Louise mentioned, this is where we got excited about discovering these chromatin marks involved in gene regulation. And so we try to figure out what are they doing. Well, indeed, there are proteins in the nucleus that have acetyl-lysine binding domains. And what does that do? Like a nucleosome remodeling complex binds the histone tail that's been acetylated. And the histone tail is part of a nucleosome. So the nucleosome remodeling is now introduced to its substrate, increasing the rate of chemistry. I'm sure many of you know this amazing story about molecular glue degraders. The origins of this comes from Handa et al., who discovered the target of the IMiDs, the drugs thalidomide, lenalidomide. That protein is cereblon. That got a lot of us, I include myself, years later, following up on this work. And in fact, 5 labs in different sort of pairwise collaborations published 2 papers back to back 4 years later, showing that what this compound is doing, not only binding cereblon, but cereblon is its presenter and it's inducing neoprotein interaction. Cereblon is part of an enzyme complex that ubiquitinates whatever's nearby, and this induces these new proteins to be ubiquitinated, and then substrates for proteasome-dependent degradation. So natural, not natural in this instance, these drugs are working as a molecular glue degrader. Now, later, Christina Wu, a colleague of mine at Harvard Chemistry Department, was studying an old, well-known, 40 years post-translational modification where certain unstable proteins have asparagine and glutamine side chains that will cleave the amide backbone of the protein into two pieces. No one knew why would that happen? Well, she figured out that inspired by the structure of the product, the C-terminal imide looks a lot like those drugs. And sure enough, that C-terminal imide post-translational modification renders it a binder to cereblon. And now the rest of the enzyme ubiquitinates this fragment of an unstable protein. So it's a protein homeostasis clearance mechanism. So in the light of what I just said, same slide, I'm going to change the language. What we have is a glue is inducing one PTM, ubiquitination, by mimicking another PTM, the C-terminal imide. To me, reinforcing this notion that there's a common molecular mechanistic theme here. Recently, a group has discovered another natural product that binds the same FKBP12 protein. In this instance, it glues it to a different target. And I give this example to start thinking about how glues can be used in various ways, because from the crystal structure, you can see the target, it's a protein CEP250, but what's amazing is that it's binding an otherwise featureless coiled coil, a classic feature that one would say is difficult to drug. It'd be hard to find a chemical that could bind high affinity and specificity, and yet, when you combine the small molecule with a protein presenter and you get that cooperative interaction, you can now bind coiled coils. Recently, the company Revolution Medicines has repurposed the cyclophilin presenter protein. They were interested in targeting a mutant allele of KRAS. So they put an electrophile on their compound that would engage the nucleophilic cysteine in this mutant allele. And then using protein structure guided design, they optimized the small molecule protein 1, protein 2 interactions and protein-protein interactions. It's a beautiful study, and there's new clinical data from this very promising compound for treating pancreatic cancers, a major unmet medical need. Another topic I want to share with you, which I think is one of my favorite sort of 1, 2, or 3 future directions, are intramolecular glues. So how would you inhibit a molecular machine called the inflammasome? Doesn't have an enzymatic activity. It's a machine that rotates through multiple conformations. This group, Carey and Decker et al., found a very simple synthetic compound that's shown. And the quote is their words. It binds 4 different subdomains within a knock domain of the inflammasome. And it glues it, as their word, they call it an intramolecular glue, locking the protein in an inactive conformation. So this molecular machine is glued in an inactive conformation. To me, the sort of granddaddy of them all, because there's a growing number of cases of this sort, is this amazing story out of Vertex Pharmaceuticals of a drug called Trikafta. This was a 30-year ordeal. What we knew about cystic fibrosis was that it's a Mendelian disorder caused by missense mutations in a single gene called CFTR. In fact, the most common allele lacks the phenylalanine at codon 508, shown in the middle panel. And this causes the protein to adopt a different shape, and it loses 2 critical functions. So how did they solve this problem? They found 3 hotspots in CFTR. They found different chemicals that bind each of the hotspots rather weakly. They found 3 that bind synergistically. Each time a new one enters, the affinity is increased. Put all 3 together, you get super high affinity binding. And then lastly, this intramolecular glue glued together different parts of the protein, returning it to the wild type structure, compensating a missing amino acid. And today, cystic fibrosis, that used to be a terminal disease, is now a manageable one for people who take this Trikafta drug. So I think this just gives you sort of the possibilities for the future. And then finish up with some combining of these two. So the phosphodiesterase family, many of the themes I mentioned, there's 2 phosphodiesterase family members. They have a very similar hotspot, 2 different chemicals binding to that same hotspot, or one in one protein, one in another protein. In one instance on the top, that creates a neoprotein interaction with a ribonuclease called schlafen 12. Normally it doesn't interact, but it gets glued together. And it actually activates the ribonuclease, which is the basis of its cytotoxic activity, and this compound is in clinical trials for treatment of certain cancers. And then the bottom one is, the compound is a drug called rolapram. It binds a similar hotspot, but in a different phosphodiesterase. And now it takes a domain that you can't see in the back and folds it over, locks it into place like an intramolecular glue, causing it to adopt an inactive conformation. And then I think my last example here is go back to that cereblon case, the IMiDs, the drugs, thalidomide, for example. Beautiful work from Gabriel Lander has illuminated the multiple steps involved in making an IMiD like thalidomide. So 1, you have to bind a hotspot, always the same in cereblon. 2, you have to be an intramolecular glue and take that domain to fold over to another domain. If you get that far, now you're a candidate for being an intermolecular versus intermolecular glue, bringing in substrates to get ubiquitinated and degraded. So that's how the simple IMiD drugs actually work, intra and intermolecular gluing. Okay, I said I'd like to then turn to this second topic of bifunctional compounds. Now, Louise alluded to this, thank you. I was collaborating with my good friend and one of the most creative scientists I've ever met, Jerry Crabtree, using these glues to study signal transduction. But we thought, well, as I mentioned in my introduction, this is a very repeating theme. Over and over, signals are transduced by protein glues. Like, what if we could make chemical glues and chemical-induced proximity routinely? So we thought, well, that's going to be hard. Each one is idiosyncratic. So we came up with a simple workaround. We said, why don't we just find a binder to one protein, a binder to another, maybe make a couple different linkers, but have a limited little assembly of these bifunctional compounds. And then we would take these 2 proteins and treat them, small little proteins, as domains, and we would just genetically fuse them onto any signaling proteins we wanted, and then have a single chemical that could induce proximity. And we say, how far can we take chemical-induced proximity as a means to intervene into biological processes? By the way, some of you may have heard of the term optogenetics. Optogenetics, and this is a nice little story. Karl Dieseroff figured this out. He was an undergraduate, and he was taking a class with me about, I'm just talking about what I'm talking to you, and he walked away and realized that, well, I know of a protein called cryptochrome 2, and it's a monomer, and you can induce it to become a dimer. But now you don't need a chemical, you use a photon. And so he took those and he genetically fused them onto all kinds of signaling proteins. That's optogenetics. It's really the same principle that I've been trying to share with you. So we've made a lot of systems, studied a lot of, this was the first time in my lab we used this to turn on a biological process with spatial and temporal control with a chemical. So we made a mouse, a transgenic mouse. We caused a signaling protein called FAST, the FAST receptor. We took just the signaling domain of it, linked it to an FKBP, and we used a tissue-specific promoter to express this only in thymocytes. We made this animal, and it's a completely uninteresting animal. It had nothing different about it relative to the wild type, except when we treated it with a chemical we call FK1012. This came from a postdoc in the lab, Steve Diver, and he took the FK506 molecular glue and ran an olefin metathesis reaction of that allyl group, and he made FK1012 out of it. That's why we got the name, double of 506. This prevented calcineurin from binding, so it's no longer a glue, but it's a bifunctional compound. And you'll notice in this transgenic animal, we've equipped it with an FKBP. So sure enough, when we treat this animal, otherwise innocuous animal, with FK1012, a single dose, we induce the proximal interaction of the FAST receptor, which is a protocaspase. It cuts itself, activates the apoptotic signaling pathway. And we could achieve cell ablation with spatial and temporal control in this manner. And this works in humans. Jerry and I started a company called Area Gene Therapeutics that collaborated with Bellicum Pharmaceuticals. They made this synthetic compound that's functioning like FK1012. And then they used this to treat graft versus host disease. So leukemia patients were undergoing bone marrow transplantation as a last step, very challenging medical process. But if you survive it, you may overcome your cancer, but you may also develop what's called graft versus host disease. So you take donor hematopoietic stem cells from bone marrow, you purify them, and then transduce these, and then, but with a certain frequency, the grafted immune system will attack the entire body. That's called graft versus host disease. And that's a lethal side effect of this process. So it's the treatment of last resort. So by transducing hematopoietic stem cells that had this same switch that I just described, they found 5 of their patients developed graft versus host disease. They took a single oral dose of remeducin, and it eliminated the graft versus host disease. As far as I'm aware, this is the first example of a bifunctional compound being used to treat patients. And then I give you this example because I mean, everywhere I go, I'm amazed at the amount of effort on these targeted protein degradation, including the use of what Craig Crews, it's part of the story I'll tell you about, calls PROTACs. The origins of targeted protein degradation are that of Peter Halley. Peter Halley is a cell biologist, and he was interested in understanding how papillomavirus causes cancer. Cervical cancer most commonly, but other cancers. And what he discovered was amazing. And it's an example of what I was telling you about before, an early example that really had this influence on thinking about bifunctional compounds. He discovered the virus makes a protein called E6. And E6 is a protein glue. It brings together 2 host proteins, an E3 ligase and P53, the guardian of the genome. So it induces the degradation of P53, a critical step for the virus to become oncogenic. So there you have it. This was another, so it was pretty simple from there to go to small molecule targeted degradation. Peter gave us the clone for E6. We fused onto it an FKBP12, and then we took various proteins of interest and fused an FKBP12, and we expressed these in cells, and we treated with FK1012, and we had targeted degradation. Now, why do you not know this story? You kind of do, and you kind of don't. You don't know it because the postdoc who did this, Martin Prushy, much to my chagrin, was whisked away by a senior executive in a pharmaceutical company here in Europe. He's done very, very well, but right in the middle of this great study. It turns out his baymate is Craig Crews. And so Craig was able to, carry the torch and has taken this in all kinds of exciting new directions for medicine using native proteins, but exact same principle. So PROTACs are simply bifunctional compounds of this sort, same principle. This company that Jerry and I started made kits freely available to academics, over 1,200 labs, and there's over 2,000 papers showing over and over all kinds of ways to intervene. So bifunctional molecular glues, chemical inducers of proximity, we have proof of concept that you can intervene in all kinds of biological processes. I take this figure from a recent perspective by Alessio Ciulli, and it really reflects this transition that I was alluding to, where we were using these tagged or fusion proteins, showing all things you can do, and then the field has moved to native proteins, and all kinds of things that have come out of this. So just in the targeted degradation, in addition to PROTACs, there's LITACs, there's all kinds of others, lots of good labs doing great work in this area. But as you can see from this review, controlling acetylation, phosphorylation, dephosphorylation, oligoside, carbohydrate transfers, which I heard some great studies of today, all kinds of interesting chemistry you can now control with chemical inducers of proximity. And my lab has been very excited about this area. I'll give you two quick little vignettes. This is work of a physician scientist in my lab, Will Gibson. He worked jointly with me and Matthew Meyerson. He's left our labs. He's joined the faculty at Harvard Medical School and the Dana-Farber. So the first thing he did was say, going back to the work with Crabtree, we showed that if you recruit a repressor to a transcriptional activator, you pick the right repressor domain, you could dominantly repress where it was activated. I've always thought this is my second favorite really new area of rewiring transcriptional circuitry. So Will was able to do this by taking an IRF1 transcription factor that has a nuclear export sequence and sits in the cytoplasm. He made a bifunctional compound that recruits a protein with a dominant nuclear localization sequence, and that shuttled it into the nucleus, as you can see in this little video. This is adding a chemical. You see what happens. That protein goes into the nucleus. But it turns out that protein has a very strong transcriptional activation effect by forming liquid-liquid condensates, among others, and indeed turns on the IRF1 transcriptional circuitry of the cell. So you take exquisite specificity of DNA binding domains of native transcription factors, but you control yourself whether you turn on or turn off. I think the number of opportunities here is really significant. A very exciting new area for medicine. Second example I'll give you also from Will Gibson. He recruited an MIT undergraduate, Ananthan Sadagopan, and in this instance, he made a bifunctional compound that has, on the one hand, the purple piece up top, which he got from a company, a biotech company called PMV Pharmaceuticals, and they had reported on a compound that binds a mutant allele of p53. Now, p53 is an incredibly important protein, not only as a guard of the genome, but roughly 50% of cancer deaths are caused by tumors with mutant p53. And there has been endless studies to learn how to target p53 mutant cancers, and no one has achieved success. Now, Will is a clinical oncologist, and he came to me and said, Stuart, in the clinic, what I do when I have surgery on a tumor, this p53 mutant, is I stain for p53, because the one thing, the sine qua non of p53 mutant cancers is there's a massive upregulation of p53. So even though he stains histologically with an antibody, it's not selective, and all cells in our body have p53, you get a super strong signal because the mutant form is upregulated 80 to 100-fold. So he said, why don't I use this as a sink, make a bifunctional compound? So he just linked this up to a general cytotoxin, in this case a PLK1 inhibitor, but I think there's a lot of different warheads you could bring in this concept. And in work that is available to you on bioRxiv, and it's in late stage of review right now, he showed indeed this compound is the first p53 selective in anti-cancer agent. We looked across a panel of 1,000 cell lines and saw that the biomarker of predictable biomarker for toxicity, mutant form of p53. Okay, now my last little section here, I want to get to this notion of most of these compounds have been discovered serendipitously, like the original mechanism of action studies. Oh my goodness, another glue. I think what we need to do is learn how to pick the presenter, pick the target, make a glue, or a bifunctional. So we started by integrating, again, something Louise mentioned, the idea of using diversity-oriented synthesis with DNA-encoded libraries. Liam Hudson, a postdoc, joined the lab and said, why don't we, rather than taking typically in Dell chemistry a single skeleton and decorate it with a lot of different appendages, why don't we use the skeleton as a diversification element? So this library that's been published, it's accessible to academics at no charge, has 70 different skeletons and then 2 appendage sites. And they're all DNA barcoded, so we can use Dell screening technology and know what the compound, or infer what the compound is by sequencing the barcode. You can see the skeletons are so different that we use rigidifying elements like bicyclic rings, bridged rings, spiro rings, fused rings, and allow us to project these appendages with different exit vectors that would be difficult to achieve with just a single skeleton. Now, very first screen using this library was done by postdoc Xuan Lu. I don't know, she was here some time ago, but I'm not sure how long ago. She was interested in a mutant allele of IDH1. It's an oncogenic form. And it wasn't for this reason, but she screened this protein, which we knew was an equilibrium of a monomer dimer. And incubate with the library, wash away, PCR amplify the barcodes, and in this experiment, barcodes 37, 31, and 120 were identified. 37 told us what skeleton, what stereochemistry. She synthesized that compound, and it was a pretty good inhibitor of mutant allele of IDH1. Then she solved the crystal structure with Michael Romanowski and a couple of other colleagues. And sure enough, what the compound was doing explained its stabilization by calorimetry. It's a molecular clamp. And it binds in such a way that it makes distinct contact with one element, one of the monomer units, the other of the monomer units, and makes extensive protein small molecule contact and induces protein-protein contacts. So without even trying, right out of the bat, a molecular clamp. Lost my track of time here. What time did we, what? Oh boy, okay. I will wrap up. We've also been learning different screening approaches. So here, typical Dell experiments take a target, a magnetic bead, incubate with library, and then compare that to the bead alone, because you get a lot of nonspecific binding. And we call that the binary enrichment. Jeremy Mason, a postdoc, had a very simple idea. Why not just do this experiment, but add a presenter protein and look for ternary enrichment versus the magnetic bead alone. And he picked a family of proteins, bromodomain proteins that are all listed there. And I'll show you just results of one of them, but it worked with every single family member. In this case, we use the VHL presenter protein as a VCB complex, a ternary complex. And the barcodes told us to synthesize this compound. It was synthesized using a nanobit assay in cells. The compound induces protein-protein interactions. And we picked this system because it's a very well-studied one in the PROTAC field. Because VHL is part of an E3 ligase complex, we expected a functional consequence, and sure enough, this compound is a PROTAC. It degrades a protein. But there's plenty of PROTACs out there. What's different about this is that we didn't find a binder and then optimize a linker. We screened a library. In one fell swoop, we got all three in a single structure. And the crystal structure showed lots of small molecule protein contact with both proteins and protein-protein. Something rather amazing that comes out of the Dell screening technique. And the last example here is work, again, of Xuan Lu. Below, you see the classic Dell. You look at the barcode enrichment under these two conditions, with target, without. Above is what I described that Jeremy did. you take the 3-component element and compare it to the bead alone. Sean had a really simple idea. Why don't we analyze our data differently and look for what she calls a high presenter ratio, which is the ratio of barcodes. I'm sorry, it's the ratio of the ternary enrichment over the binary enrichment. So intuitively, this would be the ones that confer cooperative binding. And cooperativity is the hallmark of glues. So this chart's a little bit complicated, but on that ternary enrichment axis is the way we used to do the experiment, and you get some real high signal, strong high signal outliers, and sure enough, synthesize those compounds. And they're bifunctional compounds, and they bind with no cooperativity. Over on the right in red are a series of compounds. Each diamond is a separate compound. We probably would have ignored, because it's a little bit in the baseline, but they had super high presenter ratios. And sure enough, these are compounds that don't like binding one protein. They don't like binding the other protein. But when everything is together, they bind cooperatively. That's the hallmark of glues. I don't have time to tell you all the reasons why I think for like tissue targeting, tissue specificity, systemic compounds, but will only become active when 2 pre-selected proteins are in the same location, like the disease location. And then lastly, some work that's on bioRxiv right now. We started looking at different presenters, going back to FKBP12, and completely different proteins, including this reductase, and found molecular glues from this kind of screening. So I'm very optimistic that this is a general way to discover either glues or bifunctional compounds. Sorry if I went over. Thank you for your patience. Been a delight to be here today, and I'd be happy to take any questions.
Professor Schreiber, thank you very much for a really fascinating talk, really inspirational chemical biology talk today. So we're now ready for questions from the audience and also questions from online as well. Who would like to kick us off? I mean, I certainly have a few if people want to gather their thoughts. Gentleman over here, yep.
Please.
Wait for the microphone, please, and if you can introduce yourself as well, that would be great.
Okay.
Yeah, I think so. No?
It's working. All right, great. Yeah, good evening. My name is Bohan. So first of all, thank you so much, Professor Schreiber, for the wonderful lecture. It's my honor to be here. And the question I want to ask is, so we've seen so many compounds, molecular glues, that can increase the cooperativity between proteins and biomolecules. So what's the extent to which we can manipulate exactly how much it increases this cooperativity and this binding?
Well, important question and a real hard one technically right now. We had a good discussion, I forget with whom today, about various ways to get at this. And especially with generative AI today, the idea that maybe this is something we could begin to compute in advance, or set up high throughput systems to just learn empirically. But I don't know that anyone has the answer to your question right now. It's so empirical, which is why I had to go through it very quickly. But the compounds that were bifunctional and the compounds that were molecular glues, and I use a functional definition of glues. Glues is cooperativity. It's not the nature of the structure. We can take general libraries that don't have that, and we find glues from them by this screening. But I picked those 2 because they're structurally rather similar. They do pull together the same 2 proteins. But could you ever, if I told you one's a highly cooperative, and those cooperativity factors are gold standard. They're as cooperative as rapamycin, FK506, the original glues, right? Very weak interactions, and then super strong when all 3 are together. I don't know. If you can look at those structures and tell me why, I'd be really impressed. So it's a combination right now of probably needing to do this empirically over and over, get data. But maybe in the future, although the power of generative AI and protein structure determination and design is that it works. The shortcoming is that we actually don't know how the machine was able to give us the answer. So it's a tough problem. Probably we need more data, and we need real smart people like you to go put your brains to this problem and figure it out.
Thank you so much.
Sure.
Thank you. Any more questions from the audience? Yes.
Hi, I'm Jess. I'm a postdoc at the Francis Crick Institute, also working on DNA encoded libraries. I was interested, so the work that you presented there, maybe the new work is slightly different, but the previous work that you published on, like your library is more targeted towards the
Correct.
Is your newer work like more general libraries?
Yes, yeah, I alluded to that. You're 100% correct. The sort of, we stack the deck in our favor in every single way, as you often do when you're doing a proof of concept experiment. Like, is there any hope for this idea to work? And that was the published work of Jeremy Mason and Xuan Liu. There's a company in Cambridge called Magnet Biomedicine, and they've been working hard on the problem you're alluding to. And I've done a little bit in my own lab of taking sort of, I'll call them standard. I mean, I don't think, like that first library to me it's standard in the concept, but I actually think it's got some really unique chemical features. So we're looking at lots of different libraries and have been able to find by exactly Xuan's screening mechanism. Now, more unbiased molecular glues. Now, does it work all the time? Absolutely not. Does it work half the time? Nope. It works some percentage of time. I don't know what that is. You know, maybe it's 10, 15, 20%. And so it's not a general solution, but, you know, there's a lot of problems to be solving, and I think we can live with that for a while. That's exactly what the company Magnet did. They had a number of systems they were interested in. They screened all of them, and then they basically cherry-picked the ones that had the most promising results, and they've been driving those forward. But they are really exciting examples where they pre-selected the presenter protein to be based on its protein expression. Only in the disease tissue happened to be the tumor. So, and certain tumors have very unique proteomes. So I think, you know, that's a promising direction. Only if you have cooperativity, right? A systemic drug that does nothing until it finds the right compartment where these two things exist. So I don't want to overstate that. I think it's still early, but there's some indications that standard libraries could work in some cases.
In your, like the first paper you presented, you showed about like the impact that the DNA tag can have on finding X. You put in the positive control, which you didn't pull out. So this is obviously something you've got to take into account. You've got a massive DNA tag on the library that's going to have an effect.
Yeah, if I'm understanding your question, you're saying like, well, what about sort of artifactual binding because of the DNA? Oh, it's huge. I mean, it's not just for clues, it's for all DELs. I mean, the way to think about this question is we build these libraries and we kind of dumb down the fact that there's 55,000 molecular weight, you know, piece of DNA onto a 400 molecular weight compound. It's the tail wagging the dog. And then you get something that's bound. What does that mean? It means you did that experiment where you washed away and PCR amplified. Like, why wouldn't you expect the DNA to have some role? And of course, it often does. And so it's one of these ultra high throughput, you know, the simplicity of the screen, one little tube of water, you can run 10,000 binding experiments, but it's not front loaded. The back end is, what about the role of the DNA? What about all the artifacts that come from DNA? It's a challenge. I wouldn't try to ever oversell. I mean, you know, that beautiful concept was Sidney Brenner and Richard Lerner 30, 35 years ago. And it's had some real successes, but I think, you know, like many technologies, it can work in certain instances. Yeah. Yeah, no, I love that. Yeah, all of the above. Like, you know, someone once said to me, they said something about, in a very disparaging way, about flatland. They said, oh, I'm so happy that you make these three-dimensional molecules. I'm so tired of, you know, flat sp2 rich. And I thought, you're entirely missing the point. And the point is, we've got high sp2 rich compounds. We have fewer of the more sp3, but none of that matters. You just need some of each. And then I said, if flat molecules work over and over, sign me up. My goal, the best flatland chemist I can possibly be. So metals, high molecular weight, low molecular weight. Personally, right now, I'm really kind of attracted to these very low molecular weight molecules. I mean, look at the image, look at the thalidomide. And I'm drawn to that because of the analogies, and there's just so many of them now. I just wrote a perspective that's published this year, and like the list of examples that I could have pointed to has been quadrupled since that point in time, of chemicals functioning as glues by mimicking a small little amino acid side chain. So I think it's not by chance that thalidomide is, by Lipinski rules, at least the rule about molecular weight, it's too small, right? I think nature makes jasmonic acid, jasmonate. I don't know, 200 molecular weight? How does it work in plants? How does it work? It's a molecular glue. I think a lot of low molecular weight compounds would be very appealing. But anyway, get that Gibbs free energy any way you can. And metals, that's another really untapped area. And then a few people now doing organometallic type. I mean, I'd love to see a ferrocene in there, right? Why not?
Great. We have a question online for you as well from Luca. It says, dear Professor Schreiber, what are your opinions about the peptide? It's jumped down, excuse me. Where did the question go? I'll just have a look quickly. I think the question was about the substrates being peptides over small molecules. What are your thoughts on that? And could they also be as potent?
Yeah, it allows me to go back to the unanswered part of your question. I think absolutely, why not? I mean, by the way, the term molecular glues, I wrote a perspective in 1992. I was working with Don Wiley on, he had just solved the first crystal structure of MHC protein and had the extra electron density. It turned out to be the antigenic peptide. So we now know, of course, MHCs present antigenic peptides that are made intracellular from viral infection or whatnot. And then different T cell receptors come along and they touch to see, is there something I'm familiar with, thymic education, or is it new? And what does it do? It glues it together long enough to get T cell receptor signaling. So, and it was that context. I said, look, these chemicals that bind FKBP12, by then we were seeing lots of FKBP12 glues. They're kind of like MHCs, and I called both of those. So the original term was a shout out to natural products, but also to antigenic peptides as glues. So those are linear peptides. Now, you ask about cyclic peptides. That's low-hanging fruit again. And we have proof of that. I mean, cyclosporin is just that. In fact, we don't have time to go into it, but there's so much to learn from cyclosporin, how it functions. It's got these tertiary amides. It's a super slow binder, just like antigenic peptides are for MHC. It requires a weak interaction, long enough to have an amide rotation and then a more avidity effect, and then slowly, but literally like 4-hour on rate, and very, very large, long off rate. So I think cyclic peptides, I think restricting conformation of the amidactylide by making them dialkylated, these are all theoretical concepts that have some precedent that cyclic peptides can work as glues. That RevMed example, it's a hybrid, but it's got a lot of peptidic character.
One more question from the audience.
I think cyclic peptide phage display, I've never done, but that, I'd love to go do that. I think that's a really exciting opportunity.
Perhaps I can ask you a very quick question here, and we have local schools and students here as well, and colleges. And perhaps you could maybe allude a little bit on the sort of forces that are at play for the glue part of these molecules. What kind of bonds and interactions are quite important in your research?
I think that the interactions of glues are probably nothing particularly unique about when you break them down. They're the normal, electron-electron correlation, van der Waal interactions, hydrogen bonds, all kinds of polar, nonpolar interactions. But it's just the increased level of complexity because, and we don't know the answer. There's another question that came up today I found really fascinating. It was like, is it that it binds to one protein first? In some cases, like FK506 and rapamycin, they bind FKBP12 with super high affinity all by themselves. It's only cooperative on the other side. Other cases, they're both very weak. So how does that work? I think it's a sort of standard binding interaction. In the case of cyclosporin, maybe it's really weak, but it requires these changes in the conformation of the small molecule and probably of the protein also. And then meanwhile, that same thing is going on the backside, except it's not only the protein-small molecule interactions, but now it's seeking out protein-protein interactions. So if you break them down to the individual forces, they're probably very familiar and the same. It's just the little bit increased complexity of, the timing of these events and going on in multiple ways.
Great, fantastic. Well, our time's up. Please join me in thanking Professor Schreiber one more time. Thank you. I'd like to introduce our head of department, Professor Oscar Says, to give us the final remarks. Thank you.
Thanks, Nazila. Hi, folks. It's down to me to bring tonight's proceedings to a close. So before inviting you over to have some refreshments and some nibbles, I just wanted to have a set of thanks for a number of people today. So first and foremost, I want to say a massive thank you to Professor Schreiber for accepting today's invitation, for giving just, quite simply an amazing talk, but also throughout the whole day engaging with the community. He's met with staff, postdocs, students, and he's engaged. The enthusiasm you saw here today, it started this morning with everyone, so I'd like a massive round of applause for Professor Schreiber. And then I'd like to thank everyone who made today possible. So first, that begins with Professor Wilkinson's family and friends. They've supported us over the years, and in particular, most recently, with establishing, through the Sir Geoffrey Wilkinson Foundation, the ability to support today's lecture. I'd also like to thank all the staff and students today, so that's PhDs, postdocs, and staff who met with Stuart today and engaged so enthusiastically with their science. It was really exciting to hear those talks. The students at the back here who come from local schools today to listen to the science, it's really lovely to have you here today. And again, it was great to see how you engaged. I'd like to say a massive thank you to Louise and Nazila for chairing today so well. I'd like to thank everyone else who was involved at the back end. So that's the college team that supports these events, Advancement and AV. They've done a wonderful job of bringing everything together today. And then last but not least, there's someone who's probably hiding because she doesn't like to be acknowledged in these events, but it's Sarah in the room. She's hiding. So there's Sarah Bide, who you all know really well, who's worked tirelessly for about 6 months to kind of bring everything together. Every single element of what's worked today, she's been at the heart of that. So I'd like to thank that entire community, please. Thank you. Okay, and folks, with that, what we have now is a load of refreshments available and nibbles outside. It's actually split across 2 floors. So if it does get a little bit busy downstairs, it's all replicated upstairs rather than all kind of crowded downstairs. But folks, have a great evening. Thank you. Thanks, enjoy that? Yeah. It's really good. That's amazing.